Hi, everyone. Thank you for tuning into this recorded talk. My name is Steve MacKinnon. I'm the Chief Platform Officer at Cyclica. My background is in structural bioinformatics, and the ISMB has always been one of my favorite conferences. I've been with Cyclica for 8 years now, where I currently lead a team that focuses on building new predictive drug discovery technologies, more specifically with a focus on tools that model a drug's polypharmacology. I'm here today to talk about a collaborative COVID-19 repurposing project that we participated in, where we were able to find a new host target along with some bioactive compounds that were effective across several human coronaviruses. I will be in Madison for the conference if you want to reach out to chat on site. Otherwise, feel free to reach out via the conference's instant messaging platform or anytime afterwards over LinkedIn. So this project was the result of a multi-party partnership made up of um, three primary groups, one from the Vector Institute, Bo Wang's group, who focused on the human target ID component of the project, work that was largely led by student Hao Tian. Cyclica, we focused mainly on the drug target interaction predictions in that aspect of trying to find drugs that would interact with set targets. And the work here was largely led by Dasha Redka. Um, and assay design and validation that was led primarily from Costin Antonescu's group at the Toronto Metropolitan University, but was later joined by several other Canadian labs who each offered in new assays and new validations into the study. The research was supported by a number of different Canadian funding agencies, CFAR, MyTax, and CERT, and the Toronto Metropolitan University, who each offered some funding into our two academic collaborators on this project. The work led to the publication of the paper you see right here that was released last fall in scientific reports. So if you did like this story, if you did want to cite it or read up more details about it, feel free to look this one up. DOI is listed right there, and we'll provide a link towards the end. So I would actually like to start off with the off-target drug repurposing part, and that's the part that Cyclica contributed to the project, and the part that actually came chronologically first, which we'll see and explain in just a few moments. So before we jump into the drug-target interaction predictions, I just wanted to take a moment to talk about drug repurposing strategies in general. And for that, I'd like to borrow from this review that came out a couple of years back that we really liked. We liked the way that they framed everything. And while the goal for drug repurposing is always the same, that is, for the indication that you're looking at is to find an existing FDA approved drug or something that at least has been tried clinically in the past as a therapeutic for this new indication. Um, the way to actually go about doing that could actually vary from project to project depending on the indication and the parameters of what's going on. So to start, let's first bring up the disease-centric drug repurposing, and that's where um, a new drug repurposing opportunity is proposed based off of the similarity between related diseases. And most of that first wave of repurposed molecules for COVID-19 during the early pandemic, those were already known antivirals that were developed for another virus, such as remdesivir, and just kind of knowing that viruses use RNA and to kind of block that RNA reproduction machinery through the incorporation of RNA or nucleoside analogs. Um, what we're talking about is kind of closer to target-centric or drug-centric repurposing. So target-centric is, um, so it's maybe the indications themselves aren't closely related, but they do share a mutual target. So that is the use of a shared mechanistic target um, for repurposing opportunities, or drug-centric, which that one has kind of the lowest degree of similarity between. So that is using off-target interactions of an existing known medicine to identify new opportunities and completely new indications. So that um, in that particular case, the repurposing is based off of a drug's polypharmacology. That is, not the drug's original anticipated target, but something else that the drug happens to do in a cell. And that's what we're going to be looking at today. All right, so let's get into the drug-target interaction predictions. So DTI or DTA for drug-target interactions, drug-target affinities, um, these predictions represent this emerging class of models that are trained directly on protein ligand data sets, bioactivity data sets, drug target data sets. And we've seen a recent uptick of these types of models over the last 5 years or so, mainly attributed to that increased accessibility of both the bioactivity data and the accessibility of deep learning technologies. Um, and all DTI models, at least within this class, kind of operate in a similar way of training a neural network with a pair of feature vectors, one representing ligand representation and one representing protein representations. And what they kind of all have in common is having those two paired inputs, and then training some sort of a discriminative model for either making interaction like calls, that is saying yes they interact or no that they don't interact, or potentially trying to predict affinity from that. That being said, where these models tend to differ the most is in the representation themselves. So Matchmaker is Cyclica's solution to the DTI problem. It's a global drug target interaction prediction model trained on 3 million paired bioactivities that we developed specifically to predict drug polypharmacology. So when we first started looking at this problem, um, we weren't too convinced with the protein representation. That's the part that we thought was really lacking in previous solutions. That is, when we look at these large databases like Chembl or Binding DB, we will know that this particular drug and this particular protein interact. There'll be a bioactivity associated with it, but we don't really know where on the protein that interacts with. Proteins can be much larger than the ligands. They can have multiple domains. And if we really want a good representation of the protein in order to learn patterns from, we really wanted to get that down to the binding site, because the binding site is where we would see those patterns emerge that could pair up with ligands. Say the number of hydrogen bond donors in the ligand, the number of hydrogen bond acceptors in the pocket, large pockets versus large ligands, and so forth, right? So we really thought that biophysically that compatibility should be coming from the pocket itself. So what we developed is a bioinformatics pipeline there to take those 3 million paired bioactivities in these large databases and map those onto 3D structures using a combination of algorithms and alignments and superpositions, everything that we could to try to get or try to gauge that most likely site. So we would be taking information from homologs and what homologs are known to bind ligands, where they're known to bind ligands, what kind of ligands, and use that information to map these 3 million DTI pairs onto pockets so we could use pocket features to train our DTI models. So in that sense, Matchmaker is trained on pose-independent protein ligand feature pairs. And computationally speaking, it's very fast. Screening a molecule across the whole human proteome takes just a fraction of a second. That's really what allows us to do what we call these 2D screens, where we look at a large library of compounds screened across the whole proteome. For more information on drug target interaction predictions in general, there's a link down here. It's a reference to a review that we published late last year on DTI models. So as I mentioned, DTI models require feature vectors to represent ligands and proteins. Ligand features that we use are a combination of things that describe the biophysical properties of a ligand, the size, the shape, the flexibility, or the composition, certain key functional groups, how many there are in the ligand, which functional groups exist, as well as some substructure fingerprints that are fairly standardly used across cheminformatics approaches and ligand-based bioactivity modeling in general. On the protein side of things, we do use some global protein features, much like all of the other DTI models that have been described in the literature so far. Things like domain compositions, enzymatic reactions, and select protein annotations that kind of describe the overall protein. But really where Matchmaker comes in and that angle that it provides that's different are those local pocket features. By mapping all those DTIs onto individual pockets, that's when we're able to do something similar to the biophysical descriptors of the ligand, to do that on the protein pockets. So that is, the chemical compositions of the binding sites, sequence information around the binding sites, functional groups that exist, and other things to describe the binding site geometry, the size and shape, the flexibility, and so forth. All right, so Polyform DB is the database, a precompiled repurposing database that we designed um, on the basis of matchmaker predictions. So that is running matchmaker's proteome screening on all molecules that had been mentioned in clinical trials databases. At least at that first pass, we were able to find around 2,000 molecules that were approved, another 2,000 that are in clinical trials but not yet approved, um, and a number of other preclinical molecules or nutraceuticals. Since then, we've revised the way that we create our list for repurposing, and we'll often go directly off of vendor data sets, so we know that the molecules that come up in the search can actually be sourced, which is certainly an important component of projects like this one. On the proteome side, we have a proteome of around 8,000 proteins, 8,500 proteins from the human proteome. That number has actually since increased as well as we've brought in protein structures from the AlphaFold 2 proteomes that were offered by the EBI and DeepMind. And we pull those together, we do 2D all by all molecules by all proteins, and created a database with that. Right. So right at the onset of the COVID-19 pandemic, around January timeframe is when we started seeing sequences show up in databases. People had started to isolate and sequence the COVID-19 virus. Around February is when we saw, as the virus was spreading, a number of groups were doing homology models of those viral proteins. Um, Shortly thereafter, we started taking those homology models, doing systematic pocket detection and featureization of the pockets so that we could include them alongside our human proteome and rebuild a version of this Polyform DB, that was specially tailored to start looking at COVID-19 repurposing. That being said, at the time, that's all we had really. But we were missing a key part. We didn't really know what we should be targeting other than those viral proteins. So we released this on ChemArchive, believe that would be early April 2020. And that was kind of a call to action out to uh, possible partners, anybody who might be interested in um, trying out new target ID or testing out predictions. At that time, there was a lot of government grants that were being announced, a lot of people interested in doing what they could or reallocating some of their lab resources so that they could help study the virus or help contribute to the research. So around that time, we were also really busy looking for partners, see who might be interested um, in taking this forward with us. All right, so that leads us into the target ID portion. That is the the portion of the study which is picking which targets we should actually be looking at for off-target repurposing. This particular portion of the project, the computational work of the development of the target ID graph convolutional network, was largely led by Bo Wang's group at the University of Toronto and the Vector Institute. While the kind of process of looking at the targets that were coming out, matching them with Polyform DB, assembling that, and finally getting to that list of compounds, that was done by Dasha Rudka, an applied scientist at Cyclica. So this is the high-level overview, the figure one from the paper, that shows the workflow of how we combined the target ID portion with the off-target prediction portion, and specifically the steps that were taken to get that down to a list of compounds that we could order for follow-on testing. So looking at this top portion, we have the interactome, which describes all kinds of interactions between drugs, functional pathways, human proteins and viral proteins, um, interactions between the different groups, interactions within those individual groups, um, and how that massive network is used to train a graph convolutional network, and the outcome of that particular portion being both the identification of repurposing drug candidates directly, as well as the identification or the proposing individual targets that could be related to COVID-19. All right, so getting that multi-scale interactome together for the sake of this particular project was a combination of merging previously described interaction networks, one that took the compounds, functional pathways, and human proteins that had been previously described, so a base network made up of those, as well as at the time newly released viral human protein-protein interactions from the Gordon data set that came out around April, May 2020. Those were combined together and adapted for training this graph convolutional network. Um, so having a large network like this and looking for these non-obvious associations within the network can be kind of difficult. If you were to go about doing this without that ML angle, the question then becomes, well, how would you weigh in a particular path going from COVID-19 to an individual target? What is the role of an individual protein on that path? What is the role of a known protein-drug interaction? How do you weigh those individual interactions, or how do you weigh those individual proteins in creating your path? So that's really what that network is trained to do. It's trained to basically come up with functional distances between different units within it. And the consequence of that is being able to create proximities or direct or indirect proximities between the virus, the COVID-19, and a list of different targets or a list of different existing FDA-approved drugs. So that brings us to our first set of 10 compounds that were ordered for experimental testing. And those came from direct network recommendations from the GCN. That is based off of evaluated proximity distances between COVID-19 and the drugs that existed within that multi-scale network. Starting with a slightly longer list, applied scientists at Cyclica started sub-selecting from the top, ensuring that what we were looking at were actually FDA-approved drugs, that they were small molecules and procurable for that matter. That is, we weren't going to go after nutraceuticals or the biologics in this particular study. And we want to maximize the selections for diverse chemistry and indication, given the limitation and experimental throughput. So we wanted to, increase our odds at potentially finding something with new biology. That being said, there was a limitation to this approach. It was limited to known reported drug-target interactions. And the data set in particular was pretty stringent on its drug-target interactions, having kind of a limited amount of bioactivities within it. Um, and if we just kind of think of how many human proteins actually have reported bioactivities, it's still a small fraction. So it's possible that these weren't necessarily the best targets that the network could come up with, but they were the best targets that had known drugs for them. Right. So the second thing we were looking at with the GCN were evaluated proximity distances between COVID-19 and targets within the network. And that's what allowed us to broaden the set of targets that much. It wasn't otherwise restricted to only those targets that had primary drug targets. And at this particular point, we sub-selected it down to 10 targets. So this part as well required a degree of sub-selection or curation from our expert applied scientists within Cyclica, who looked at the longer list of proteins or ranked ordered list of proteins and started sub-selecting them based off of three different criteria. We were looking for proteins that were present in Matchmaker, that is, so that we could do the cross-referencing with our off-target interaction predictions. Those that were not linked to adaptive immunity alone, so we didn't want anything that would show up in that GCN target ID because of its downstream effects. Um, these weren't things that the functional assay that was being developed by our collaborators would be able to measure. So targets that we suspected might be involved in that were removed. And we also wanted to look for new things altogether and off-target interaction predictions. So we did exclude molecules that already had FDA-approved medicines. All right, so that's where we recombine the uh, 10 targets that came through the target ID GCN back with Polyform DB, our repurposing database of cross-screened human proteins versus clinically tested drugs. And at that point, we kind of also picked a few based off of uh, different selection criteria. One selection criteria where we're just looking at single target, we were trying to find compounds that had good single target ranks or proteome screening ranks relative to the targets that were identified by the GCN. And then another one, we were looking for compounds that were ranking multiple targets fairly highly, just on the off chance that that could potentially be better as well. So to elaborate on that a little bit, the top 10 targets coming out of that GCN, we picked the top ranking compound out of Polyform DB for each of the 10. Then we also looked a little bit more into this multi-target selection, where we would identify compounds that out of those um, top 10 targets might have two or more of those targets ranked pretty well. Um, and then from that set of 35, looking at redundancy, applying some, diversity metrics, we kind of narrowed that list down to 16. So a lot of the compounds that did well on the multi-target task also did well on the single target task and vice versa. And a lot of those 25 were similar to one another. So altogether, 16 compounds that were selected in the hybrid approach, where the targets were identified by the GCN and the compounds themselves with their off-target interactions predicted through Polyform DB. And that adds to the other set of 10 for a total of 26 compounds that were ordered for follow-up testing. All right, so now on to experimental validation portion. As a computational person, this is where kind of our role started to taper off, and our experimental collaborators started to take on all the heavy lifting. And this part was largely led by Costin Antonescu's group out of the Toronto Metropolitan University, formerly Ryerson, where at least at the beginning of the project, Um, First few weeks following the initial shutdown, they had developed new viral infectivity assays to be able to screen compounds or screen the predictions that we were making. And then later on, as we were getting some interesting data, he brought in a number of other collaborators from his university and from other groups within Toronto to kind of help add more context, add more validation into this, and pull the whole story together. I'm honored to be able to present this on their behalf, but just that note there that much of this work came from experimental collaborators. All right, so on to data. Um, So our collaborators have developed this immunofluorescence screening activity that looked for the coronavirus spike protein, not on SARS-CoV-2, but on another coronavirus, the 229E, that is endemic in humans and responsible for kind of the one of the common cold viruses. This one in particular didn't require the biosafety level 3 labs and could be conducted in, I believe, a level 2 safety lab instead. And given that the purpose here was looking at um, viral host targets, things that potentially multiple coronaviruses may share in common, we were hoping that we could first screen on this and then look at what the activity might look like on other coronaviruses. So what the assay is doing is it's measuring how much of that coronavirus spike protein is in a sample after a fixed amount of time, I believe in this case 48 hours after infection, um, in the presence of a given drug and without it, right? So all of these numbers are relative to a baseline in which that particular drug is not present. That being said, it's not shown here, but the assay was also calibrated using remdesivir, one of the nucleoside analogs, and it showed something quite similar to these two cases right here, the capmatinib, and the other one over here that showed highly reduced activity in the presence of the drug. That being said, 4 out of the 26 had some degree of measurable activity, which is quite remarkable, given that in a lot of these cases, we are simultaneously predicting targets and off-target interactions. So we were quite pleased to see this particular hit rate. And one thing of note is that these three compounds actually did have this IRAC4 compound in common. So for at least the remainder of the study, the compound that we looked at the most was this capmatinib, mainly because it had one of the most potent activities, and um, its at least predicted off-target interaction was the same off-target predicted interaction as a couple other ones on this list. So that's the one that we followed up on for the rest of the experiments that we're going to present here. So these two plots show follow-up from that previous screening hit with capmatinib on the 229E coronavirus. The one on the left is the same assay that was performed on the screening, except in this particular case, it's showing that the antiviral activity is operating in a dose-dependent manner, meaning at 10 micromolars where we see kind of that 95% activity range. When that drops down to 1 micromolar, we're getting to maybe a third of the activity. 100 nanomolar, even weaker, down to 0. So that's the plot on the left. The plot on the right is showing kind of a secondary assay that was done. This is plaque forming units, so it's a different way to kind of measure how much virus that it's seeing, and it's showing with this second assay, same thing, that capmatinib is active in the 10 micromolar range. So moving on to other coronaviruses, these are results for the NL63 coronavirus, which is another endemic one that is sometimes responsible for the common cold. In this particular case, we're also observing a dose-dependent behavior with capmatinib around the and a good 90%-ish inhibition at the 10 micromolar range. That being said, in this particular case, it's measured on the plaque forming units, but it's also kind of confirmed by looking at RNA levels as well, viral RNA loads. So now we're on to our third endemic coronavirus, and that one is the OC43. In this particular case, capmatinib is also showing antiviral activity, at least with this plaque forming unit assay. That being said, it's perhaps not as strong of a signal as we showed in the last few slides with the other coronaviruses. That being said, it's another thing to note when looking at this PFU data. The PFU data, at least in this chart on the right, is operating based off of counts, how many plaques are being, and that to some extent could be under-reporting antiviral activity, because in reality, it's not just the count, the size of the plaques might also convey a degree of antiviral activity. So but perhaps the numbers aren't as sharp as the other two, but it's still pretty clear that there's some antiviral activity going on here as well. All right, so now on to the more impactful coronaviruses. That's the SARS-CoV-1, SARS-CoV-2, responsible for the 2002 SARS outbreak as well as the COVID-19 pandemic. And again, for safety purposes, these assays were conducted with pseudoviruses and an inhibition assay to measure infectivity of these pseudovirus constructs. And just for clarity, those pseudoviruses are these assemblies that are made up of a combination of the full-length spike protein for the respective virus. So that is, the SARS-CoV-2 full-length spike protein, along with a lentiviral backbone and a luciferase reporter. Those are combined to create these virus-like particles that are exposed to cells, and if they manage to enter human cells, then the luciferase reporter will show activity. So, using these virus-like particles or these pseudoviruses, the collaborators were able to measure capmatinib's antiviral activity, and again, kind of getting to the same micromolar or 10 micromolar range as was observed in those other endemic coronaviruses. So, having shown that capmatinib had antiviral activity across multiple coronaviruses, the next step was understanding why, and to see if that mechanism was indeed in line with the combination approach from the target ID GCN and Polyform DB that led to it being brought up for the screen or being a candidate for the screen. So that being said, first step was to check to see if its antiviral activity had anything to do with its primary target. So capmatinib was originally developed as a metastatic non-small cell lung cancer therapeutic, specifically targeting the protein tyrosine kinase MET. To check to see if this MET activity had any relationship to the antiviral activity, a second MET inhibitor, AMG 337, was also tested. In this particular case, the alternative MET inhibitor, AMG 337, had no antiviral or anti-coronavirus activity. So together, that's implying that capmatinib's antiviral activity is likely MET independent, that is off target. So another note there is that this AMG 337 is chemically distinct from capmatinib, and that chemical distinction is what's likely driving that differential polypharmacology, right? So even though they could both share one target in common, the fact that these molecules are different is likely means that they would have different off target interactions. So the target that was linking capmatinib to the coronavirus in the whole GCN and Polyform DB workflow was IRAK4, another protein kinase. So in order to see if that could be part of the mechanism, um, capmatinib's antiviral mechanism, the collaborators ordered this tool compound JH125. It's a known dual inhibitor of IRAK1 and IRAK4. It showed similar activity profile to capmatinib in both the plaque assays and the immunofluorescent experience. Um, Secondly, collaborators were able to show that an IRAK1/4 siRNA knockdown into cells, even without the compounds, without the JH125 or the capmatinib, the um, the IRAK1/4 knockdowns were also equally resistant to infection. So right there, that's some convincing evidence that there is a role for IRAK1 and IRAK4 within the antiviral or within the virus infectivity pathway. All right, so that wraps up the presentation and the bulk of the content that came from this joint publication released last fall. The full reference is down below if anybody wants to dig a little bit deeper, perhaps check out the code bases. Feel free to look us up. In summary, we've presented the discovery of a rational drug-centric COVID-19 repurposing candidate. We were able to show computational technologies that simultaneously predict the new targets and some salient bioactive compounds that had that anti-coronavirus activity. So in that sense, kind of this full stack machine learning prediction. Um, we were able to demonstrate primary repurposing candidate capmatinib had efficacy against multiple coronaviruses, and that's likely due to its host-based target. Capmatinib's antiviral activity was not driven by its primary target. It's that is MET, so it was acting in a MET-independent manner, in that its antiviral activity was the consequence of off-target interactions. And lastly, that IRAK1/4 inhibition protects human cells from viral infection. So, stated differently, kind of on the biology side of things, that IRAK1 and IRAK4 are likely involved in the cell infectivity pathways. So yeah, so thank you for listening into the entire recorded talk. Uh, as I mentioned earlier, if there are any questions, feel free to look for me in Madison if you're attending the conference in person. Otherwise, reach out via the conference's instant messaging platform or on LinkedIn afterwards. Um, so if you did like this presentation and you like hearing these drug discovery stories, do look us up on our website, cyclicrx.com, on the slide, Twitter or LinkedIn. Reason being is our team puts out a lot of um, seminars, symposium. We have a molecule to medicine series and fireside chats in which we talk to drug discovery experts in the industry with kind of informal interviews or more seminar style talks that describe different parts of the drug discovery life cycle. So if you did, enjoy hearing about this story, do connect with us, and we do have a lot more stories to tell. Thank you.